Human reproduction relies on two biological objectives: generating specialized gametes and executing their targeted delivery. In males, this operates as a constant, unceasing factory. While ovum formation in the female reproductive system ceases naturally around age 50, the male system is built for endurance. Gamete production here can persist well into old age. Maintaining that level of lifelong cellular output requires a highly specialized, multi-stage anatomical pipeline. This macroscopic pipeline is located primarily within the pelvic region. The entire system is organized into three integrated tiers. First, the primary organs, or testes, handle manufacturing. Second, a symmetrical routing network of accessory ducts manages transport. Third, specialized glands provide necessary chemical support. To understand how these tiers operate together, we have to trace the precise spatial route of a single gamete from its microscopic origin to its final exit. The male reproductive system functions as a precisely timed logistics network. Every structure along the route exists to solve a specific physiological constraint of gamete viability. The genesis point for all male gametes is the testes. In human adults, each testis is an oval-shaped organ, roughly 4 to 5 centimeters long and 2 to 3 centimeters wide. Notice their placement. They sit entirely outside the primary abdominal cavity, suspended within a pouch called the scrotum. This external location solves a core environmental constraint. Mammalian spermatogenesis simply cannot occur at the standard internal temperature of the human body. This chart shows the narrow viability zone for sperm production. To yield functional gametes, the environment must be maintained exactly 2 to 2.5 degrees Celsius lower than core body temperature. The scrotum acts as a dynamic thermoregulator, physically expanding and contracting to perfectly maintain that exact thermal deficit. Because of this exposed position, each testis is wrapped in a dense outer covering, shielding the delicate architecture inside. Placing the primary manufacturing centers outside the body core creates structural vulnerability. That vulnerability is the required evolutionary trade-off to satisfy the strict thermal parameters of continuous cellular synthesis. Moving inside the testis reveals a highly compartmentalized interior. Each organ is divided into roughly 250 distinct compartments, known as testicular lobules. Inside each of these 250 lobules are 1 to 3 highly coiled seminiferous tubules. This is the actual physical site of sperm production. Scaling down to the microscopic level, we can examine a cross-section of a single seminiferous tubule. Lining the inner wall of the tubule are the male germ cells, or spermatogonia. These primary germ cells undergo a series of meiotic divisions, eventually transforming from parent cells into fully mature, independent spermatozoa. But the germ cells do not operate alone. They share the tubule with a second critical cell type, the Sertoli cells. Operating as nurse cells, Sertoli cells span inward from the tubule wall. They provide direct, essential nutrition and physical scaffolding to the dividing germ cells. Gametogenesis is a highly codependent mechanical process. The germ cells rely entirely on the structural and nutritional support provided by the Sertoli cells within the isolated environment of the tubule. Outside the seminiferous tubules, we find the spaces filling the gaps between the coils. These are the interstitial spaces. They contain a localized network of small blood vessels and a separate crucial cell population. The primary operators of this extratubular space are the Leydig cells, also known as interstitial cells.
Leydig cells synthesize and secrete the testicular hormones, known collectively as androgens. They share this space with other immunologically competent cells, which provide a localized defense network for the testis. The physical creation of sperm happens isolated inside the tubule, but the biochemical trigger for that process, the androgens, must be continuously manufactured and supplied by the interstitial engine on the outside. Once spermatozoa are manufactured in the tubule, they face a logistical challenge. They have to be transported out of the extra-abdominal testis and back into the body cavity. Gametes exit the seminiferous tubules into the ray testis, traveling upward through the vasa efferentia to breach the testis boundary. They arrive at the highly coiled epididymis on the posterior surface, where they are stored and mature. As the route continues, the tightly coiled epididymis straightens out, transitioning into a thicker duct known as the vas deferens. The vas deferens ascends vertically, leaving the scrotum to enter the abdominal cavity, where it loops entirely over the top of the urinary bladder. The primary function of these interconnected ducts is to safely store and transport the maturing sperms across the physical divide of the pelvis. The ductal network acts as a sequential biological highway. It bridges the physical gap between the low-temperature manufacturing site outside the body and the internal delivery mechanisms. Spermatozoa alone are in T viability and motility. They require a specialized biochemical vehicle. This vehicle is added at a convergence point, where the vas deferens receives a duct from the seminal vesicle, merging to form the ejaculatory duct. 3 accessory glands operate along this junction: the seminal vesicles, the prostate, and the bulbourethral glands. Together, the secretions from these glands deliver a critical fluid payload known as seminal plasma. This diagram outlines the biochemical composition of seminal plasma. It is rich in fructose to provide immediate energy, alongside critical concentrations of calcium and specific enzymes. In addition to these nutrients, the secretions from the paired bulbourethral glands provide critical targeted lubrication to the penis to facilitate direct insemination. Following the addition of the seminal plasma, the ejaculatory duct opens into the urethra. The urethra extends directly through the specialized erectile tissue of the penis, terminating at its external opening, the urethral meatus. The accessory glands transform raw cellular products into a complete biological delivery package, ensuring the gametes have the necessary resources to survive outside the male body. The male reproductive system operates as a single, synchronized manufacturing and logistics pipeline. The entire process rests on a chain of strict dependencies. Without the precise thermoregulation provided by the extra-abdominal scrotum, cellular meiosis fails. Without the interstitial androgen engine working on the outside, and the structural support of the Sertoli cells on the inside, germ cell differentiation stops entirely. And without the sequential routing of the ductal network and the biochemical payload provided by the accessory glands, gamete delivery becomes impossible. This specific anatomical arrangement guarantees the continuous, viable production of gametes throughout the human male lifespan. The biology of human reproduction relies on exacting structural engineering. Every macroscopic loop, gland, and pouch serves to protect the microscopic demands of cellular synthesis.